All right, so sorry about the technical difficulty there. But as I was saying, what I was going to do is give a little bit of a historical perspective and discuss some of the points that Gerhard brought up, because that seems to be a lot of interest to the group at large. And so what we're going to talk about briefly is a slide made some time ago by Professor Merrill about why we want to look at lipids and why they're important, because they are key regulators of cell structure and cell function. And what they do is they control things like the energy, the structure, transport of molecules into and out of the cell, as well as cell signaling. And then their implications are when they're not in balance, we can have either excess or deficits of energy. We can have aberrant sterile cell function. Transport mechanisms can be thrown off, and we get differential signaling as well as developmental abnormalities. Now, some of the fundamental questions we ask as scientists are, what is it? Is it a phospholipid? Is it a single lipid? Is it a cholesterol ester? What is it that's changing? The second most important question is, how much is there? Is it micrograms? Is it nanograms? Is it picograms? What is it? And how does it change over time? Does it go up? Does it go down? Is it unchanged? Or is it there a flux that goes through a particular pathway? And then finally, we saw in the first talk, where is it? And moldy imaging happens to be one of the very interesting things that's come about in the last 5 or 10 years. And so when one thinks back historically about the classical methods of lipid analysis, things such as GC or HPLC or TLC, typically they required chemical modification procedures for detection. However, these could introduce artifacts. They required large quantities of sample to go into the machine or onto a TLC plate. Or they lacked specificity with regard to the head group, the fatty acid, as well as the unsaturation, not only the number of unsaturations, but where they were located and whether they were cis or trans. So, being that I'm giving a talk on lipid maps, a long time ago at a hotel far away, a group of lipid researchers gathered to discuss forming an alliance to identify and quantify all the lipids in a cell. And there's your LipidMAPS team from almost 20 years ago. I believe you already heard from Bob. I think you heard from Ed and Al. And here I am, as usual. I'm on the outside, I'm the outlier, being the least experienced and least intelligent one of the group. And so that's when we decided to dive into lipidomics, right? Where we wanted to do a comprehensive study of the metabolism and pathways of all the lipids in a given biological system. And so what we want to do is preferentially isolate and enrich those lipids via an organic phase extraction. And then we typically analyze these lipids by mass spectrometry. And the reason we do mass spectrometry is because of the very high sensitivity of the instrument. Also, we get a high degree of specificity with regard to mass, and when we do MSMS with regard to structure. And when one uses MRMs, we can quantify these species very rapidly. Okay, and so in our discussions in lipid maps, we decided that what we needed were really good internal standards, because at the time, these standards simply did not exist. Typically, a researcher would partner with a synthetic organic chemist and have that chemist make molecules for them. And so it was rather hit or miss whether or not you were really being accurate. And our internal standards are in essence a molecular yardstick that we use to measure how much of the analyte is there. And so we use these internal standards, mass, when we do MSMS for structure, when we use LC, retention time, and as you also saw earlier in the previous talk, drift time for ion mobility. Okay, and we use these internal standards for quantitations, as Gerhard mentioned earlier, to control for extraction efficiencies, whether or not everything goes into a particular organic phase. We use them to control for ionization efficiency, as he also alluded to earlier. So we want those internal standards to be in the same droplet as our analyte of interest, and also for fragmentation efficiency. So the number of double bonds that are in the lipid chain. And also, and this is a kind of a subtle point, it's a quality control check that we use to demonstrate that our instrumentation is functioning properly. Okay, so if we don't see our internal standards, then we know something is either wrong with our chromatography or something is wrong with our mass spectrometer. And so we know to stop and check and see what's happening. And so as we sat down and discussed what we wanted for our internal standards, we came up with several criteria. And it was mentioned earlier, what you want is the internal standard to have the same chemical and physical properties as your analyte of interest. Ideally, this will be stable isotope labeled analogs, right? So that way they would co-elute or co-ionize. However, that was not always the case early on in LipidMAPS, because what, at least for the sphingolipids we chose, the route we chose, was either short or odd chain species because they were more cost effective and they were easier to produce, right? And so ultimately what we want is our internal standards to have utility for omic type analysis, meaning we want multiple internal standards to be in our sample, so that way we can quantitate each of the individual molecular species present. However, it was not possible to synthesize all of these isotope labeled internal standards in a cost or time effective manner in the context of LipidMAPS originally. And so as Gerhard alluded to earlier, there are also some additional considerations for internal standards. The deuterium labeled analog should contain enough heavy atoms to be observed distinctly from any endogenous analyte's isotopic envelope, right? So you want it to be shifted away from that envelope so you have a clean signal relative to the analyte you're measuring. The deuterium should also not be in an easily exchangeable position, such as a hydroxylation or an amine or an amide, which would cause the internal standard to decrease in quantitative value. And if we're performing tandem mass spectrometry, ideally the fragment ion should also contain the deuterium, so you can easily distinguish it from the analyte of interest. And it should also not be involved in the fragmentation process, also muddying the waters with regard to quantitation. And as also mentioned, our short or odd chain species should not be endogenously present, as that would also throw off a quantitative value. So in LipidMAPS, it was, as most people know, an NIH-sponsored glue grant that was funded for 10 years. As a result of that grant, Avanti made approximately 75 quantitative internal standards that were designed and synthesized using the nomenclature shown there. And I actually saw, looking at the people that are present, one of the key people in making that happen was Dr. Xiangrong Li, and he's present in the session today. So if there are any questions regarding that, he might be able to answer those for you. And over the course of the 10 years, there were over 50,000 vials of standard provided to the LipidMAPS group during that grant period. So needless to say, the R&D team was very busy. And the LipidMAPS internal standards were really a key revolution in lipid quantitation, because they opened the doorway for us to have the ability to quantify individual molecular species for many researchers around the world, not just for LipidMAPS. And so as Gerhard alluded to earlier, the general workflow starts either with a large population group or two sample sets, say a normal versus an altered. This could be a disease state, this could be treated with an endogenous drug, or it could be a knockout mouse, something that's going to affect the lipids that are in a particular cell or tissue. You simply spike your sample with an internal standard cocktail, and knowing how much is there to begin with, but you know how much internal standard you need to add. So scanning your particular tissue or cell of interest prior to putting the standard in is going to be helpful. Generally, what we find is if you're within 2 orders of magnitude of the endogenous level, be it higher or lower, your calibration curves are very good. So what we do then is extract both our analytes and our internal standards. We concentrate them, dry them down, and resuspend them in our solvent of interest. And then we do MS analysis followed by data interpretation. And here's just a simple example of the early days whereby we had a single internal standard. We use multiple chain links of ceramide or dihydroceramide, and we plot those across a concentration range to get a straight line. And as you can see, the original things that we had analyzed all lie pretty much on the same line. So to begin with, we had very good quantitative numbers. And what flows from that is the ability to measure many different lipid species simultaneously. And so we get a very complex data set that comes from that. And this is just a simple representation where one can see, here we have sphingosine being acylated, going to ceramide and so forth, going down to sphingomyelin, to glucere, ceramide phosphate. And all of these things can be measured in one run. So now I'm going to skip over to some do's and don'ts, some tips and tricks that we've learned over the years regarding lipid quantitation. And the first one is to read the literature first, because somebody somewhere out there has probably tried to do what you're doing on an instrument similar to yours. Okay, so they may be right, or they may tell you what to avoid, which will save you time, effort, and frustration. We get quite a few emails from customers that try our standards and say, oh, they don't work on my machine, or I don't see this particular lipid, or I don't see that particular lipid. Generally, the instrument manufacturers make their machines to their specifications, and lipids typically aren't on their radar screen. And so the other one that we want to point out is don't start immediately with complicated analytical techniques such as LCMS or LC-MS-MS. Not all mass spectrometers are the same. How ions are formed and transferred makes a huge difference. Some instruments have skimmer cones, some instruments don't. Some instruments have heated metal capillaries, some instruments have glass capillaries. Knowing your instrument is going to be key to getting the best performance out of your internal standards as well as your analytes. You probably want to infuse your sample and optimize your mass spectrometer first to see what species your instrument is able to observe, and also at what level you're able to observe it at, because some machines are more sensitive than others. And it's important to remember that ions come in two flavors. They can either be positive or negative. If one side isn't working, try the other. And some species, such as neutral lipids, may need some help. That means adding some type of modifier adduct, be it a salt, ammonium salt or something, or a chloride anion or something of that nature. In a similar vein, not all liquid chromatographic devices are the same, and not all C18s are the same. There are differences in injection volumes, sampling ports, washing and rinsing cycles, and pressure limits that can make a difference in what you see and when you see it. In a similar vein, elution characteristics will be different depending on the flow rate, the column, and the solvent system. Typically, I know for sphingolipids, we use water or methanol. We rarely use acetonitrile, but a lot of people do use acetonitrile. And this will have a different effect on where things come off the column. And C18 columns aren't created all the same. If you buy one C18 column from, say, an Avonex, it may have different elution qualities than, say, a BEH column from Waters. So you need to pay attention to what you're using. So the second thing you need to do is optimize, number one, retention on your column. Make sure everybody sticks on the column. And then elution, make sure your mobile phase is strong enough to get everybody off the column so that the species of interest are observed in your chromatogram. And it's important when you couple the two together, both the LC and the mass spectrometer, that the ion source, heat and gas and flow rate all match. That way you don't encounter either thermal decomposition or incomplete desolvation and reduced sensitivity. So that was a historical perspective on where LipidMAPS started and where we are today. And so now we're moving on to Lipid Standards 2.0. And so as LipidMAPS project started to sunset, we asked ourselves the question, what's next and how do we improve and how do we expand? So originally, the LipidMAPS project was designed for the mouse macrophage. So obviously we wanted to branch out and go to new and different biological systems that would be important to the research community out there. And so we needed new applications. And these constituted primarily human health, disease, or looking for biomarkers. And as such, we wanted to add new lipid classes, such as the plasmalogens that weren't covered originally. I think we saw earlier in the talk, the PIPs, and I know one of my favorites is the gangliosides. And as Gerhard pointed out, we needed more unsaturated species, right? The monos having double bonds in different positions, as well as the polyunsaturated species. And then what we wanted to do too was make very simple lipid standards kits that people could use, ampulated samples that you could just pop the top off and have enough internal standard to spike very large numbers of samples. And thus came up with the Splash family of standards, which is essentially a molecular toolbox. Much like you'd have a ratchet with different socket sizes, what we wanted to build was a kit of lipids that had different chain lengths, different mixtures, and different classes all in the same vial. And so some of these Splash standards we have are comprise at least 17 different lipid classes, both complex as well as the lysoforms. These are deuterium labeled. They contain between 3 and 9 deuterium atoms, and they have odd chain lengths. These, as I mentioned earlier, are prepackaged, and you can get them at different quantities. So for like mouse plasma, they can be at the endogenous level you would find in the mouse, or with the EquiSplash, they can all be at the same concentration, or they can be at variable concentrations. And what we also did was we made the individual components available so that these can be customized with regard to the class or the concentration of whatever particular biological system you're looking at. And so our new applications that we were targeting involve cardiovascular disease, neonatal testing, whereby you take a dry blood spot, put it on a piece of paper, punch out the paper, add your internal standard, and look for diseases that arise from either genetic or metabolic disorders. Also, what we do was we were wanting to look at the lysosomal storage disorders, things such as Fabry, Gaucher, Krabbe, or Niemann-Pick type A or B. And you'll notice all of these are associated with sphingolipids. You can see the ceramide, the glucose, the galcer, and the sphingomyelin that are implied there. So more recently, our collaboration with Chemicruz, we came up with what we call the ultimate splash. And this particular splash mix has multiple deuterium chain length variants within 15 different lipid classes. And this again is what Gerhard alluded to earlier, right? And what we have are many different unsaturation variants within those 10 different lipid classes. So hopefully this will address some of those problems. And also there are variable concentrations within each class. And there are approximately 70 different individual molecular species in this particular mix that are designed such that their M/Z occurs between the naturally occurring lipids and their fragment ions are unique. And so as was alluded to earlier, what we wanted was for this particular mix to be multi-technique applicable. So that means with regard to sample introduction, you can use it either in a flow injection mode, or you can use it in conjunction with chromatography. It also means that we can use it with normal phase or reverse phase. So you don't have to worry about where things elute in reverse phase and differences in ionization. We have different chain links covering the different elution times. And with regard to mass analysis, we can use these particular standards either with targeted methods, such as the shotgun methods, where we do precursor ion scans, neutral loss scans, MRMs, or things of that nature. And they can also be used with untargeted methods, such as ultra high resolution accurate mass measurement. And so this is just a typical certificate of analysis that shows that the particular internal standard comes in solution of DCM with methanol, contains about 1 ml of your standard, a little bit more. We like to overfill a little bit. And they're simply stored in a freezer and they're good for a year. And so there are also little tips here about making sure that you warm it up, sonicate it, and make sure everybody gets in solution before you use it. And the typical sample size will be about 10 microliters. So out of a 1 ml vial, you can get about 100 samples analyzed. And so, as again, I keep mentioning Gerhard, he stole a lot of my lightning with regard to the discussion of internal standards, but there are differences in ionization efficiency that results in differential signal response. And that's shown in this slide here, where one sees the different calibration curves depending on chain length and the number of double bonds that occur in a particular lipid species, in this case, phosphatidylserine. And so here, this is just a typical example of what's included in the ultimate splash. You can see the different triglycerides here. We have start with the fully saturated species here. And as we go down, we add units of unsaturation and more units of unsaturation in different chain lengths. Now we have unsaturation in different locations, and then multiple unsaturations, as well as 2 double bonds in this particular thing. Also, you can see what we've done is we put different concentrations of these different lipids into the mix. And so what this looks like on your mass spectrometer is something like this. So for the sphingolipids, they have a little bit different biological abundance. Typically what you see for the sphingolipids is high abundances either with the short chain lengths or the very long chain lengths, and in between not so much. Phospholipids typically exhibit a different behavior where we see this roof-like effect where you start low, go high, and then it comes back down low again. So what we've tried to do is mimic what's out there in biology. Now, briefly, I know there have been a lot of questions that people have been asking. And so we're going to kind of take a little bit of a side trip and discuss the elephant in the room with regard to Avanti. So as everybody that's listening to this knows, 2020 was quite a challenging year, and it was quite challenging for us as well, because the main thing we had to deal with was COVID. But the second thing we had to deal with was CRODA. And so briefly, with regard to CRODA or COVID, in March, we actually cut our staff and began working from home. Avanti has approximately 150 employees that are on campus at any given time. When we did our reduced staffing and working from home, it was reduced to about 50. And so myself and some of my QC colleagues were pretty much on campus because the samples don't prep themselves and they don't get on the machine themselves. So we were some of the few people that were actually allowed to go out, drive around, and come to work to analyze samples and get things done. Then in May, we shifted gears to large-scale production of lipids to support vaccine development, and manufacturing began. And I'm not revealing any secrets here because it's public knowledge that Avanti and CRODA are part of the Pfizer team, and we make many of the components that go into the Pfizer vaccine. As such, many of our resources were reallocated to help this particular function. So I head the analytical services group. Lisa, my co-author on this particular talk, heads the Lipidomics group. And I mentioned Xiangrong earlier, his R&D division team. We all threw resources at this particular project. Now, with regard to CRODA, in August of 2020, we were purchased by them, and in particular, their life science sector. And this particular company is a UK-based specialty chemicals company. And so what's happened at Avanti is we've actually undergone a campus-wide expansion, including a 15,000 square foot GMP manufacturing building that's currently under construction to support more of the vaccine. And starting in January of this year, we implemented a 2-shift plan, and we're working 7 days a week. And we're interviewing and hiring for a bunch of new positions on campus. So if you need a job, please see Lisa and arrange an interview. But right now, our mantra on campus is to keep calm and keep making lipids. And that's what we've been doing all along. And so I'm going to tease a little bit. And again, Gerhard, thank you. What's coming out soon? I saw Al's on the line as well. We have a single splash 1 and a single splash 2. These are deuterated, odd chain dihydroceramides and ceramides, the glucosyl ceramides, the galactosyl ceramides, lactosyl ceramides, as well as sphingomyelin. And the splash 2 contains deuterated forms of sphingosine, sphingonine, and their associated phosphates. And here's the one I was alluding to earlier. Xiangrong has made approximately 40 or more bile acids. These are both labeled and unlabeled. He has the glycoforms, he has the taro forms, as well as the other different isomers. And then, also mentioned previously, we're coming up with a multi-splash that's specific for imaging mass spectrometry and other MALDI applications. I think I saw Tucker on the line, and Tucker might be able to address these if there are any questions regarding that. And we still have other goodies that are still on the synthesis drawing board. So there's still more to come, and we're still working. So briefly, in summary, there are many different lipid internal standards that are currently available. We have multiple classes, multiple chain lengths with both fully saturated, monounsaturated, and polyunsaturated for quantitation. We have single-use vials that can facilitate large numbers of samples using various types of mass spectrometry systems as well as techniques. And we also provide individual components or custom mixtures should you need them, or should you have a unique biological system that you want to investigate. And so if you need a custom quantitative mass spec standard, just let us know. You can contact us at the Lipidomics at AvantiLipids.com link, and that will go directly to Lisa. And you can talk to her about what you need with regard to the lipids, the concentration, the solvents, or what size package you would like. And so with that, I'll conclude and take any questions.